Okay, now, the next thing. What these guys do. All right. For the most part, the wind signaling plays many different developmental roles. I think probably the biggest kind of thing would be helping to determine the dorsal ventral axis of embryos. Not just of embryos, but also of various kinds of organs and tissues. If you want to generalize the roles of the Wnt proteins, that's probably about the best way you can do it. Is the Wnt proteins, Wnt signaling leads to belly. Yeah.
Why did you write ventral and then write under it ventral?
Oh, because it stimulates, because Wnt signaling, when beta catenin goes in, it generally leads to transcriptions of genes that are going to push tissues and stuff into ventral type structures. In other words, a lot of it like gut, a lot of the endoderm, more of the endoderms in the ventral part of the embryo, and likewise ventral structures of various kinds of organs and tissues. So, in other words, if you don't have Wnt signaling, what's going to happen is what predominates is the more dorsal and ectodermal and dorsal mesodermal type stuff. As a matter of fact, that's what was going on with our sea urchins. If you block Wnt signaling completely, and that's nickel chloride, I think it might actually directly attack the Wnt protein. And the heavy metals like nickel are general toxins to all kinds of proteins. But anyway, what happens there is you're not getting Wnt signaling anywhere in the embryo. So what happens is, okay, the default effect is to form dorsal, more ectodermal type structures. So it forms it throughout the entire embryo, and all you get is a fuzzy, ciliated ball of cells. Now, on the other hand, if you activate beta-catenin throughout the entire embryo, and that's what lithium chloride does because it shuts off GSK3 throughout the whole embryo, then that kind of overrides the default of going into ectodermal dorsal type structures, so everything turns into ventral mesoderm and endoderm. So you end up with all gut and not much of anything else. Okay, so that in itself illustrates something really dramatic. The importance of this Wnt signaling pathway in very early in embryonic life, it's saying, what is the dorsal ventral axis of that? Now, we saw that in sea urchins. That can also apply in regular other invertebrates. Now, sea urchins, of course, are more closely related to chordates than anything else, and it certainly happens in chordates. In frog embryos, once again, what you're going to find out is a lot of Wnt signaling occurs over in here, and this is going to be the ventral structures of the future embryo. You know, by the time you get to the embryo here you go. Here's your little kind of yolk plug, not yolk plug stage, but sort of the early neurula. And guess what? This is belly. It came from here. And that's because Wnt is getting activated here. Beta-catenin stays, goes into the nucleus. So we're seeing this kind of thing, and it happens in a broad range of organs. That also illustrates something. This type of signaling, which we see in vertebrates and invertebrates, things like arthropods at least. I don't know if it's been found in worms or anything like that. But certainly in arthropods as well as in chordates. Now, arthropods and chordates separated from each other probably at least 550 to 600 million years ago. And the first multicellular animals are suspected to be about 700-ish million years ago from genetic and some fossil evidence. So in other words, very early on in the proto-animals, they already had some version of the signaling pathway to tell them what's up and what's down during development. And both invertebrates, like arthropods, and chordates use the same pathway in slightly different ways. And chordates, especially higher vertebrates, have a lot more versions of the Wnt proteins. So we can do a little bit more specialization. So that's a big kind of thing. And we see a whole number of different types of different winds that can cause all kinds of effects if you have various types of things. But let's take a look at an example of what some of the winds, there's many different winds involved, but what some of the winds do. Some of the winds, at least one of them, is involved in things like kidney differentiation, forming the kidney tubules and the little machinery of the kidney, what they call the glomeruli, that actually do the blood filtration and stuff. That you can thank some of the winds for that. Several winds are kind of breaking the usual rule that they can also play a role in the anterior to posterior differentiation of the neurotube. In other words, we end up with things like, okay, what's going to be the brain and what's going to be the spinal cord? Some of the winds, there's other proteins involved too that do this kind of stuff, but some of them are going to do that. Winds, along with things like fibroblast growth factors and some of their distant relatives and some of the TGF-beta superfamily members, they play roles in things like the somite differentiation. Not any single one of them has a massive, defining role here, but they all play roles in things like that. So once again, the somites, what are they going to form? Well, parts of them are going to form what we sometimes call the axial skeleton. In other words, the rib cage, the spinal column, and don't forget, the spine is not from the notochord. A lot of people did that. The notochord disappears, and the spinal column, the bone, forms around the neurotube. The notochord is underneath the neurotube. Okay, so, all right. A lot of people do that. Okay, notochord does not come from the spine. All right. Okay. Well, at any rate, so the somites, well, once again, your axial skeleton, basically your ribs and your spinal column and things like that. Limb bones and stuff, that's different. The fibroblast growth factors and other things play roles in that. But anyway, you got that. And also the dermis of the skin and a lot of the muscles of the skeleton, the rib muscles, the back muscles, and things like that. If you happen to be a biped, then the back muscles get very sore after one reaches a certain age. Okay, so at any rate, that's another kind of thing. So they play lots of roles in the spinal differentiation. They can also do things in limbs. They play some roles in limb formation. In this case, mostly dorsal ventral axis of limbs. Okay, now, you can do things where they do gene knockouts in mice and stuff like that with various Wnt proteins. There's probably about a dozen or so different Wnt proteins in mammals, each having their own developmental role. But you can do things like do knockouts of specific Wts and get grossly abnormal kidney formation, or you end up missing things like parts of the brain when you get rid of other Wts. Or you end up with things like foot pads on both sides of your paws. Yeah, dorsal ventral axis. Ventral, dorsal. Thank the Wts for that. Okay, so you can do all kinds of things. Gene knockouts of various kinds of Wts have done a number of different types of defects depending on the individual kind. So clearly the Wnt proteins play very important roles in a lot of developmental events. Especially, of course, helping to set the original dorsal ventral axis of the embryo itself. And like I say,
what little we may have gotten from last week's experiment can illustrate dramatically just how important wind signaling through beta-catenin really is. It really can screw up the fundamental development of an embryo big time. And then they die and rot. And stink.
And smell.
And stink, yes, I know what those things smell like, I've seen it, they're pretty nasty. Okay, well, that's pretty much about it, this is actually a good time to hand things, we're pretty much about it over here. This is a good time to hang it up. We'll deal with hedgehog.